Hello, welcome to the 2018 Nutritionist webinar. I'm Marianne Fessenden from AMTS and your English language host. This monthly webinar series is dedicated to providing technical talks from internationally recognized educators for listeners around the world. Paula Turiello from Córdoba, Argentina translates and hosts the Spanish language webinar. Marcelo Hentz Ramos from 3R Lab will join from Brazil, translating into Portuguese, and Tom Long from Hemingway in China will host in Mandarin. There will be a question and answer period immediately following the presentation. Listeners can submit questions through me, Marcelo, Paula, or Tom. A complete recording of archived webinars as well as the question and answer session for each will be available on the AMTS website. For those of you who like to listen to presentations while driving, we have converted the videos into MP3 files that can be downloaded to your device for offline listening. We're very pleased to host Dr. Laura Hernandez, who is an associate professor in the Dairy Science Department at the University of Wisconsin-Madison. Her area of research has focused on how serotonin controls the mammary gland and various aspects of lactation. Dr. Hernandez combines basic research from the cell to whole animal level in a variety of mammalian species to broaden the focus on the importance of the mammary gland and its contributions to and the regulation of successful lactation in dairy cattle. The outcomes of her novel research are aimed at demonstrating the presence of factors, specifically serotonin, produced within the mammary gland that can control animals' physiology while lactating, particularly during the transition period when cows are most metabolically and physiologically challenged. She focuses on the interactions of serotonin and calcium metabolism during the transition period. Today's presentation will focus on the role that serotonin plays in calcium homeostasis during the transition period. And I'm super excited about it. When I talked with Laura yesterday, we had a really interesting talk about the cross-species type of studies that she's doing in lactation. So I want to thank you, Laura, for joining us. I know our audience joins us in anticipation of your presentation. To that audience, if you have questions that occur to you during the course of the presentation, please type them in the chat window or the question and answer window. We'll answer them at the end of the presentation.
Okay. All right. Well, thank you so much for giving me the opportunity to speak, and I hope you all find my research interesting. And as Marianne mentioned, we work on serotonin in the mammary gland, and as of late, we've been really focused on the interaction of serotonin and calcium, particularly in the transition dairy cow, as this is a time when she's very challenged. So just to talk a little bit about calcium status at parturition, particularly during early lactation. But we do know that all mammalian species are challenged by their calcium status during early lactation, and this is because calcium is the primary mineral in milk. Now, dairy cows are unique clearly because they make quite a bit more milk, which means there's a lot more calcium going in than a lot of other species. But things to note of importance are that during late pregnancy, calcium is lost from the blood to the fetus at a rate of about 80 milligrams per kilogram, and this is corrected on a metabolic body weight. But what's most interesting is that this changes to about 500 milligrams per kilogram at the onset of lactation. So this is where you see a very rapid demand put on the cow's body to get all this calcium into her mammary gland to make milk. And during a day, a cow will deplete her calcium blood levels from 20 to 30 times per day in order to make enough milk calcium. And so the other thing that cows are challenged with that doesn't necessarily happen in other species is that cows don't have as great of a bone turnover ratio at the onset of lactation. And it's this bone pool or this bone store of calcium that we'll talk about in a second that the animal is going to be really reliant on to help compensate for this drain of calcium going into the mammary gland and her inability to keep up with that just solely based on what's provided in her diet. And because dairy cows are completely milked out, they're going to lose calcium much more quickly than other species because we are completely removing their milk a number of times during the day, anywhere from 2 to 3 times, but maybe even more depending on your system. So if we look at this in terms of breaking this down more basically, calcium loss associated with pregnancy and lactation, you look at this on a grams per day basis. In the growing fetus, this is 3 to 6 grams. Making colostrum, this increases up to 23 grams. And early milk, this is 30 to 50 grams per day. And we hit peak milk, this is about 80 grams per day. And if you think of this more practically, if you think about one piece of chalk here, there's about 5 grams of calcium in one piece of chalk. So if you look at this and you think about a cow hitting peak milk, making 120, 130 pounds, maybe even 160 pounds of milk per day, we're talking about an entire 12-piece box of chalk plus 4 more sticks of chalk that she's having to deal with going just into the milk. And a cow is just simply, and any other animal really, it's very difficult to keep up with this solely based on just what's coming from her diet. So why is hypocalcemia a problem? Is hypocalcemia a problem? We actually have relatively low incidence of what we would typically think of as clinical milk fever in the United States these days. It's pretty well managed. We don't see a lot of our typical down cows from this, but you can see anywhere around 2 to 5%. I have heard that these numbers are higher in Europe or in pasture-based systems where they simply don't have the ability to manage their diets as well as we can in a total mixed ration system. And if you're looking at the Merck Veterinary Manual, clinical hypocalcemia is going to be anything under 1.4 millimolar, and anything in the subclinical range is 1.4 to 2.0 millimolar. However, I will note that recent data over the last 5 or 6 years, and these are very large data sets that have come out of Canada and Florida and other places, suggest that this cutoff for subclinical hypocalcemia actually might be 2.1 or 2.2 millimolar, and this depends really on what outcome variable you're looking at. Displaced abomasum, ketosis, inability to breed back. The papers are different depending on that. But these are the Merck Veterinary Manual cutoffs. And if you look at Tim Reinhardt's paper from 2011, using these cutoffs, you have anywhere from 25 to 50% of the United States dairy cow population falling into a subclinical range postpartum. Now, what does this mean? Now, if you're in the clinical range, you're talking about down cows, and that's clearly an animal health and an animal welfare problem. We don't want to see this. But the other part of this is that may not be manifested or seen on a dairy farm is the subclinical disease and what the implication might be on DA rates, ketosis, retained placenta, mastitis, among other things. Now, this monetary breakdown hasn't been done in a while, but if we look at the vet bill and the associated loss in milk yield, this can range anywhere from $150 to $300 per case. And if you do some numbers, if there's 9.2 million cows in the United States, and that's going to be roughly $900 million. And in the state where I'm from, where there's about 1.3 million cows, this could be a $125 million problem to the dairy industry alone. And if we talk about a 120 cow farm or so, which would be the average size in Wisconsin, would be about $12,000 annually. But this does not take into account anything beyond the vet cost and the loss in milk yield. And as I mentioned, some of this data that has come out over the last several years suggests that subclinical hypocalcemia could be leading to other issues. So the cost could be quite a bit higher. Now, if we look at hypocalcemia, this just means low calcium. And the primary things that are going to be affected by this is smooth muscle function or muscle function in general. Muscles absolutely require calcium, as a fuel to cause contraction, and it's part of the cellular process related to that. Now, downstream of where can you find smooth muscles in an animal and that are clearly important for a dairy cow, this is going to be the rumen, the gastrointestinal tract, the uterus, and as well as the teat sphincter within the teat itself. So what does this mean? This means that there are a lot of downstream effects that if you don't have enough calcium within the cow to maintain her own bodily functions, you can have a whole host of other effects occurring. And we think of hypocalcemia sometimes as an iceberg effect. So if you have a defect in rumen or GI tract motility, in uterine and muscle contraction, teat sphincter contraction, you are potentially increasing your risk for a decrease in feed intake, which can lead to issues with energy balance. It can cause the rumen to fill, leading to DA. The more thing that we've seen of late are these associations of subclinical hypocalcemia and reproductive dysfunction, and a lot of this work has been done by Jose Santos' group at the University of Florida, and an increase in retained placentas, decreased pregnancy per AI, as well as reduced fertility. But also what's really important that has also come out of Dr. Santos' lab is this effect on immune function. And this was also seen by others at the National Animal Disease Center in Iowa. Tim Reinhart, Jesse Goff had shown that calcium is really critical for immune function, and that's because immune cells use calcium as a way to mediate their actions. So if we have issues not only with muscle, but we also don't have enough calcium available for the immune cells to function properly, and we also know that early lactation cows already have depressed immune function, we can have other consequences of hypocalcemia directly. And this can lead to reduction in pregnancy rates, longer intervals to pregnancy, as well as production losses, among other things that have yet to be fully understood. But we have had quite a bit of great data come out in the last few years from several groups showing that calcium is a lot more than just what we used to think of, which was a down cow clinical milk fever. So how is calcium regulated during lactation? Normally, calcium, as you can see here on this left side of the slide, you take calcium in, your gut is going to absorb it, and it's going to regulate the serum calcium levels. And that's typically going to be regulated by uptake in the gut, absorption of the calcium by the gut, and that'll primarily be regulated by excretion through the kidney. Usually in normal situations, you're not going to have a lot of skeletal calcium driving this system unless you're in a very strange instance. Now, during pregnancy, this shifts some because you're dealing not only with the cow or the human's ability to deal with calcium, but you're also dealing with a growing fetus that's having to deposit calcium in their skeleton. And so, in addition to taking and increasing absorption of calcium at the gut, which you will see, and that's going to hopefully maintain the maternal serum calcium concentrations, as well as seeing more going into the renal excretion route, you're also going to have a lot more transfer of calcium to the placenta. You will see a little bit maybe coming from the skeleton, but still most of the driving factor of calcium regulation during pregnancy is going to be this intestinal absorption. And if you think back to a couple of slides before, the demand that the fetus is placing on the mom is far less than what we're talking about during lactation. So during lactation, you are going to be taking calcium up at the level of the intestine. However, this is not enough no matter how much we feed an animal. There's a point at which giving them a limestone block or dumping a bunch of more calcium carbonate in the diet, they just can't get enough calcium to compensate for what's going into the milk. And that's because you go up drastically in the demand for calcium placed by the mammary gland. So you have a lot of calcium going to in this case, this is a human diagram, so you see breast milk, but this would be milk. But what's driving this is going to be largely resorption of calcium from the bone. So there's going to be an increase in bone turnover that occurs during lactation that is really critical to maintaining the serum calcium level that is going to be different than what we see during pregnancy or during normal physiology in an animal. And this is where a lot of our research has been focused. So if we look at, should we prevent hypocalcemia or treat hypocalcemia? I'm fully of the belief that prevention is key, and there's going to be a lot less loss on the cow in production, as well as less exposure or risk for other diseases if they don't have severe hypocalcemia to begin with. So a couple of things that are utilized commonly, most commonly, is the use of a negative dietary cation anion difference in the prepartum. And as you can see here, this is the common equation that's used. It's a predictor of successful application of a DCAD diet. And this can be done through a variety of different supplements. There's lots on the market, plus there's a lot of their own different nutrition companies will make their own anionic salt supplement. And this is going to be done for about 21 days prior to calving. However, this can become an issue sometimes in a one-group housing system, and whether can you use this for the whole prepartum period or not, and there has been research done on this. And to date, it really is the most successful strategy used in the dairy industry. There's also the use of low calcium diets prepartum, and this makes use of the common negative feedback regulation of calcium. So calcium, like most metabolites, you get too much, you're going to have a signal that drives the stopping of production. If you don't have enough, you're going to drive signals that produce more calcium or release more calcium. So the premise of a low calcium diet is that if you get below 20 grams per day, you can draw on this stimulation to pull more calcium from bone. However, this is quite difficult with current feedstuffs, particularly alfalfa, to get to this low dietary calcium enough that you can get it low enough to drive this response. However, on the market, particularly, there's been quite a bit of this done in Europe, are the use of clay binders, and there are people doing research on this in the United States now to drive calcium low in this pre-fresh ration. Now, in terms of prevention postpartum, a lot of people are using calcium boluses, and these can be given prophylactically. And these are used a lot in the industry. However, recent data out of Jose Santos's lab, and I think in others, have shown that this is not nearly as effective as maintaining blood calcium. And you need to give quite a number of these boluses to keep the calcium where you would want to. The other challenge with this is that you're going to have to be giving these orally, and with multiple boluses being needed, the ability to get the cow in and do that might be a challenge as well. They are, however, if you use them in combination with a negative DCAD diet, I've had producers tell me, and nutritionists, that if they're maintaining their DCAD really well, they might give boluses to their third lactation cows that are really high producing cows, and that really keeps them in better shape when doing that. So this can happen, and if you know that your cow is at risk, so if you have animals with hypocalcemia, you definitely want to go ahead and give them boluses as well, but they typically work better if you're also managing your prepartum diet as well in combination. So how does DCAD work? Now, the basis of a DCAD and whether you should be negative or positive and how negative should you be is based on some data that shows that if you have a very negative DCAD diet, you're going to have less risk for milk fever incidence. And that's what this graph would show here. You have DCAD on your x-axis, and then on the y-axis you have urinary calcium excretion, and on the other Y is your milk fever incidence. So you can see as negative DCAD or DCAD gets more positive, you increase your risk for milk fever. And as your urinary calcium decreases, you have an inverse relationship. Now, another set of data that has gotten, and this has become quite a hot topic as of late, is if you're feeding a negative DCAD diet, should you feed high calcium or low calcium, or what level of calcium should you feed in combination with the negative DCAD prepartum? There are two meta-analyses that have been done. The one here on the left was the first one published by Dr. Gary Etzel, I believe, in 1991. And it was comprised of about 1,000 cows. And then the one on the right was published in 2006 by Ian Lean, Pete DeGaris, and Elliot Block, and it's simply adding about another 1,000 cows to this graph here on the left. So it was just taking advantage of more data that had been published. Now, what you'll notice here is this typical bell-shaped curve that we have, and on the x-axis we have percent calcium in the prepartum diet, and on the y is incidence of milk fever. Now, what I want you to notice is that what you can see here is that you have tails. So there's this range of calcium, and we have a tail on the left that ranges from 0 to about 0.5% of dry matter. So you would need to take into consideration too how much the cow is actually eating. But you can see there's this one tail here where you have this very low calcium, which is very intuitive and kind of falls in line with that with the low calcium diets that I mentioned before. But what we also have is this other tail, where we're about 2 to 2.5% of the diet calcium. Now, I will make mention here that this data set, these values out here on the tail have been calculated largely based on the data that is present that the authors were using to make these calculations. So these bell curves were generated using data that was published, and then they made calculations to try to get a best guess estimate of what you should have in the diet along with the negative DCAD. What I will bring your attention to that's kind of interesting here is that the increase in predicted milk fever incidence is highest here in this 1 to about 1.5% range, where you would have a higher risk of milk fever. And a lot of our diets are somewhere in this 0.8, 0.9, 1% range, and we might be actually formulating diets where we're not in exactly the right range of calcium in combination with a negative DCAD, but more actual data exploring this experiment need to be done. So why is calcium so important? It's a mineral, it's really small. Typically we like to think that glucose might be more important, and glucose is really important and very homeostatically regulated, but calcium is even more tightly regulated than glucose is. It's the key mineral in milk. It's going to be excreted freely, but it's also going to be excreted into milk along with different proteins and anionic carriers. The other thing that makes it really important is that it's very important intracellularly in signaling. So it's needed for a lot of processes that we don't think about at the whole animal level, but more at the cellular level. And one thing I want you to keep in mind as we progress through this is that the main regulator of ionic calcium concentrations in the body is this thing called the calcium sensing receptor. And it's present on all tissues, on the parathyroid gland, in the gut, and in the mammary gland. And it's really going to be able to detect the ionized calcium in the blood and tell the body whether it needs more or less calcium. And that'll become important in a second. So I'm not going to spend a lot of time on this. It's just a cartoon to show you that basically movement of calcium in the mammary gland is very complex. So this is the mammary epithelial cell. This is the blood side of the mammary gland. This is the milk side of the mammary gland. There's the calcium sensing receptor I just mentioned, and there's a lot of different molecule transporters that are going to move calcium into the mammary gland, that are going to put it onto proteins to be excreted, that are going to keep it inside the cell in the endoplasmic reticulum and the Golgi, and that are also going to actively pump calcium out of the mammary gland as well. So there's a lot going on just in the mammary epithelial cell alone. And a lot of what we've seen in our system is directly related to these pumps. So I just want you to kind of keep your eye on the calcium sensing receptor, this one transporter ORI1 and then this pump, PIMCA2, that moves the calcium through the tissue. Now, if we look at a cow, so this is calcium flux during lactation, she's got her extracellular calcium pool, which is about 11 grams of calcium, and then she's got 3 to 3.5 grams in the blood in the ionized form. The cow's skeleton has anywhere up to 8.5 kilos of calcium present. So a dairy cow is going to mobilize up to 13% of her bone mass just during the first 30 days in milk. So this is a lot of calcium coming from the bone. You're also going to have active transport of the calcium being prevented or being excreted more into the urine. You're going to have an increase in passive transport in the gut to pull more calcium into the gut to absorb it into the blood. And then you're going to have a variation in dietary calcium depending on where you are at in your ration or stage of physiology. But you're going to have this flux of calcium going into the mammary gland, and as we saw earlier, this will increase when you hit peak lactation, and all of this is going to have to be maintained over and over and over again during the cow's life. So calcium is very dynamic, and there's a lot of different sources coming in, but during lactation, we're going to be relying heavily on the skeleton to keep up with the cow's ability to make milk. So how are the systems connected? They have to be able to talk to each other. And during a normal physiology that we saw in a few slides before, parathyroid hormone is the main hormone that controls calcium in the body in a normal, non-lactating animal. Now, during lactation, the mammary gland does something what we like to call sometimes is act as an accessory parathyroid gland. And it will start to produce this hormone, parathyroid hormone-related protein, which is very closely related to parathyroid hormone. They work through the same receptor. However, PTHRP is not dependent on vitamin D like PTH is, and PTHRP is not made in the parathyroid gland. And the only time you can detect it in the circulation is during lactation. And in humans, you can also detect it when you have cancers that will go and metastasize to the bone and start chewing it up. But primarily, it's only going to be detected in normal states would be lactation. So what we know in terms of how calcium is going to be moving through the mammary gland during lactation is that calcium is going to move into the mammary tissue. So this is our mammary epithelial cell here, this purple box. Calcium is going to move through this ORI1 that I mentioned previously. It enters the tissue. It's going to go into various spots within the mammary epithelial cell itself, but it's also going to get actively pumped into the milk, and it's also going to get secreted with the milk protein. And as a result, that's going to decrease blood calcium concentration. So that's seen by these smaller calciums. Now, this is then detected by this calcium sensing receptor that says, wow, we have all this calcium going into the mammary gland and the blood levels are getting low. Now, once that happens, this calcium sensing receptor will stimulate PTHRP production by the mammary epithelial cell. And then this can then enter the circulation of the animal and turn over the osteoblasts and the osteoclasts and increase calcium release from the bone tissue. And that's going to help bring blood calcium concentrations up. Now, we believe that serotonin is regulating this system in a couple of different ways that I'll show you in a second. At the calcium level, at the level of PTHRP, and potentially also directly itself at the level of the bone. So what is serotonin? Serotonin is basically a modified tryptophan. So it's derived from the amino acid L-tryptophan. The rate-limiting step is converting L-tryptophan to 5-hydroxy-L-tryptophan through this enzyme tryptophan hydroxylase. And this will become important, this molecule here, because we make use of this in all of our studies. And then 5-HTP is then converted to serotonin. But basically, serotonin is a modified amino acid, and we can readily use 5-HTP to manipulate serotonin concentrations. And 5-HTP is only converted to serotonin, whereas tryptophan can be made into a lot of different things. So what we've shown in mice, and we had to start in mice because we like to use this as a model before we take it to the dairy cow in order to know more refined information so that we can do experiments in the cow and have a better idea of what's going to happen. And what we showed in mice that on day 10 of lactation, which is peak lactation in the mouse, they have a 21-day lactation cycle. This green staining here is PTHRP, and you can see there's lots of it in the tissue. Now, a mouse that is unable to make serotonin outside of the brain, so they don't have tryptophan hydroxylase 1, they have much less of this staining. You can see there's very little of it compared to the diagram on the left. And when we gave these mice the 5-HTP I talked about, so that we'd be bypassing that tryptophan hydroxylase 1 enzyme, we see a rescue of the PTHRP concentration. So if you look here at the x-axis, hours after 5-HTP administration, we see a linear increase in PTHRP concentrations as this happens. So this led us to believe that serotonin was really critical for this axis to be stimulated. Now, when I arrived at the University of Wisconsin, I knew if this is ever going to be applicable on farm in a dairy cow, more than likely feeding it would be much easier to do manipulation of this. So we had to start in rats because we don't know if 5-HTP can actually make it past the rumen. So we fed rats 5-HTP at 0.2% of their total diet for about 20 days in this model. And what we were able to see was that their total calcium concentrations increased on day 1 of lactation if they were fed the 5-HTP prior to birth, and that by day 9 of lactation, their total calcium concentrations had actually increased. So this suggested that feeding the 5-HTP would allow us to manipulate this system, at least in a monogastric model. We also showed back in our TPH deficient mice that this was a bone mechanism. So if we look at a wild type mouse, day 10 femur, these circles here are osteocytes, and they're going to be what chews up the bone tissue during lactation. The TPH1 knockout femurs had far less of these osteocytes, and they were a lot smaller than the wild type ones that you see here. But when we gave the 5-HTP back to these animals, what we were able to see was that these were a lot larger, and there were a lot more of them present within the bone tissue when we added 5-HTP back to these mice. So we fully believe that this is stimulating more calcium release from the bone tissue. Now, what the heck does this have to do with dairy cows? I know you guys aren't very interested in mice, but they gave us a lot of knowledge before we moved to the dairy cow. And some of the things that we had to do early on was proof of concept or proof of principle. So one of the first things we did was we went out to our farm at Arlington here at the University of Wisconsin, and we took blood samples from 42 cows, and they were matched. So the farm staff identified them as a down cow, milk fever, and then they had a matched cow that gave birth that same day that they did not observe to have milk fever, and this was identified by the farm staff. And we took blood samples from these animals. And what we showed was that if we look at total serum calcium concentrations here on the y-axis and serum serotonin concentrations on the x, that there was a positive relationship between concentration of serotonin and total calcium in the cow. Now, also what we saw was that we saw a positive relationship between serum serotonin concentrations and plasma PTHRP concentrations as well. Now, I know that these are small numbers and they aren't very strong R values, but they were significant, and they were the very first time we'd actually tried to measure serotonin and PTHRP in a cow. So we were at least able to see a relationship that we had previously established in rodents. So the next thing that we did was we went out to 2 different farms, 2 commercial farms here in southwestern Wisconsin. One was a Jersey farm, and we wanted to look at what serotonin concentrations look like over the course of the lactation, and were these relationships similar to calcium in a dairy cow. Now, as you can see here, On the x-axis is day relative to parturition. So we sampled very heavily during the transition period, -5 to day 10 in milk. And then we had 5 other time points later in lactation. And this will be true on the next graph as well. The red line is serum serotonin concentration, and serum total calcium concentrations is the green graph. And what you can see here is that there's this relationship between serum serotonin and serum total calcium, with serum serotonin concentrations decreasing around parturition, that we see that with total calcium as well, and then they start to come back up as the animal goes out into her lactation. Now, we don't know if the serotonin is driving the drop in calcium or if the calcium is driving the drop in serotonin as of yet. This held up on the second farm. We also saw that we investigated this at, and this was a Holstein farm, and there was about 35 cows at each farm. And this was done by my master's student, Spencer Moore, and we still see this relationship, even though on this farm, you can see that total calcium concentrations are much tighter, but so were the serotonin concentrations as well. So we were able to see that serotonin was dynamic, and then around the time of parturition, total calcium and serum serotonin seem to be related on commercial herds. So the next thing we wanted to do was give 5-HTP like we had in a rodent model and give this to a cow and see what these different variables would, how they would respond in the blood and how it would impact calcium metabolism. And this being that this was going to be the first time we were going to do this, we had to utilize a late lactation non-pregnant model. So these cows on average were giving about 30 pounds of milk per day, not the finest in our herd. When you like to do new things in research here at the University of Wisconsin, they will give you a very basic model to start with. So this was a very proof of concept study. And this was a 4x4 Latin square. So we had a 0 milligram per kilogram dose, a 0.5, a 1, and a 1.5 milligram per kilogram dose. And these were all given IV. And this data is representative of data collected 4 days. So we gave 4 treatments, and we sampled right before the treatment, 5, 10, 30, 60, 90, and 120 minutes after we gave the 5-HTP infusion. And so these values are LS means and standard error of the means over the course of 4 days. And very surprisingly, what we found was that an immediate response to a 5-HTP treatment, total calcium concentrations go down. And I will say my grad student came into the office in a total panic, and she was pretty much convinced this was the end of her PhD, and she's like, we've lied. This is completely not what we thought. We've never seen this happen before. But we'd also never measured calcium concentrations immediately after giving 5-HTP. And if you think back to how calcium is regulated, it's regulated through a negative feedback response. And so very fortunately, we had also taken urine samples right before we started treatment and 120 minutes after the treatment. So we took one here and we took another sample here. And what we saw was that actually as 5-HTP dose increased, we saw a decrease in the amount of urine calcium excreted. And because we also had milk samples from these animals, what we saw was that as dose of 5-HTP increased, so higher the dose, the more milk calcium was put out. Unfortunately, we don't have fecal samples from this study, and we don't have any samples from the gut, but I suspect that probably there was an increase in absorption of calcium at the level of the gut as well. So this suggests that when you give 5-HTP and you increase your serotonin concentrations, at least immediately, and potentially what we think is happening is the mammary gland's taking up more calcium so that we can drive this negative feedback response and potentially increase gut calcium absorption, but also drive calcium resorption or increased calcium resorption from the bone. And so this was really exciting and kind of actually supported what we saw in our mouse data. We had just, everything we had done before had been sampled 20, 30 days after we'd given 5-HTP treatments. And at that point, the calcium levels were up to normal, and they were also in transition studies, so transition mice or rats, but we'll look at the transition cows in a second. So the next thing we wanted to test was, could we give dairy cows 5-HTP prepartum and impact calcium metabolism postpartum? And we also looked at some differences between Holstein and Jersey cows, but I won't spend a lot of time on that here today. And if you have questions about that, I'm happy to answer it. So what I want to show you here is that we started giving that 1 milligram per kilogram 5-HTP dose about 8 days prior to parturition, give or take. All our cows are on a double off-sync, so we do have pretty good handle on when the cows are going to calve, but they are biological animals, so we also can't predict if they'll go early or late. And the red line is going to be your 5-HTP cow, and the blue line are the control cows. And so what you see here is that, and these are going to be prepartum, and then over here we have our postpartum cows. And what you see is that at parturition, these cows given the 5-HTP daily for about a week prior to birth had higher total calcium concentrations. And then that these stayed higher as the cow got further out into her lactation. So day 5, day 10, out to about 12 days in milk. So actually giving the 5-HTP prepartum in these Holstein cows improved postpartum total calcium concentrations. So we were pretty excited to see that this held up as we had seen in our rodent model as well. Now, we went on to repeat this in another study. So in collaboration with a researcher in Switzerland, Dr. Rupert Brockmeyer, they repeated the study like ours, but in their system, which is a little bit different from a feeding strategy than we have here in the United States. And what they saw was here in these treated cows, as one would expect, their serotonin concentrations were higher, and that would be these open bars. And the control cows given saline, serotonin concentrations are lower. So they're getting the 1 mg per kg 5-HTP. And as we saw, you see here, their total calcium concentrations around the time of parturition was higher in the treated cows. But most excitingly, what we saw was they measured parathyroid hormone concentrations. And we had some questions about, really, was it PTHRP or was it PTH? And if you think back, as I mentioned, it's the open bars that are the 5-HTP treated cows. And actually, their PTH concentrations stay pretty steady. And these cows that were treated with 5-HTP and that had steady PTH actually had more total calcium in their serum. Whereas the cows that had lower calcium concentrations and had saline treatments, or our control animals, had higher PTH concentrations at calving, and they actually had less calcium in their blood. So this supports our hypothesis that this serotonin axis is really mediated through PTHRP and not PTH and the traditional vitamin D calcium story that had previously been reported in cows. So the next question we started getting quite a bit was, does serotonin interact with DCAD? This is something we do all the time. We utilize DCAD to manage our dry cows. So are there any relationships with this? So the way DCAD works, the hypothesis that Jesse Goff has put forth is that with this negative dietary cation anion difference, we're creating a controlled metabolic acidosis, and this allows parathyroid hormone to interact better with its receptor on the bone, allowing for bone turnover to function better, and that this has to be done with a normal magnesium level, and that does make a difference. So people were asking, does DCAD affect serotonin, or does serotonin affect DCAD? And we had no idea. So in collaboration, and now this has just come out in the last month with the University of Florida and Dr. Jose Santos, they performed a study in which they had a negative and a positive DCAD group, and we measured serotonin concentrations in these cows. And in the cows that were fed the negative DCAD diet, as you can see here, they had elevated serotonin concentrations compared to the controls. And once the cows were placed on a common lactating ration, this relationship went away by about 3 days in milk. So this prompted us to design a study in which we wanted to test whether feeding negative DCAD in combination with giving a 5-HTP treatment like we had done in our previous work, would you have a better similar or no relationship with calcium concentrations. And so what we have here is actually ionized calcium concentrations on the y-axis and days relative to parturition on the x. And you can see the key over here. So we have a negative DCAD group given 5-HTP IV for about a week. Prepartum, and the negative DCAD was for 21 days. A negative DCAD with saline, a positive DCAD diet for 21 days with 5-HTP treatments, and then you have your positive DCAD with no 5-HTP treatment or the 5-HTP treatment or control. And what you see here at parturition is that the combination diet with the 5-HTP treatment had the highest levels of ionized calcium concentrations, and the negative DCAD diet alone and the 5-HTP diet, 5-HTP treatment with positive DCAD alone were similar, and the lowest ionized calcium concentrations were in this positive DCAD without any 5-HTP treatment. And these are ionized calcium concentrations, as I mentioned, and this was just accepted in the Journal of Dairy Science a couple weeks ago. Now, when we look at total calcium concentrations, you can really see here again that the combination of the negative DCAD along with the 5-HTP treatment resulted in the highest total calcium concentrations in early lactation, not only at parturition, but on day 1 and day 2 of lactation. And the negative DCAD diet alone or the 5-HTP treatment alone prepartum had similar total calcium concentrations at parturition, and these positive DCAD cows with no 5-HTP treatment had much lower total calcium concentrations. And if you'll note back to those values before, this cow is getting, isn't quite clinical, but they are just about, they're in that medium subclinical range. These other two are actually subclinical at calving, and these were within a couple hours of calving. These samples were taken, and then the combination diet was above this subclinical threshold. So these cows would have been at about normal calcemia, if we, depending on what paper you look at. Now, what I want to show you here that was actually in direct conflict or not what we saw, at least in the Florida study, and this could be a number of reasons. Now, we formulated these DCAD diets to be approximately the same in terms of concentration. Ours were about -7. I think the Florida study ended up at about -13. We were both trying to target -15 and didn't quite get there, but we also have different dietary treatments. So there's different components of the diet in the Midwest, clearly, than what you would have in Florida, so that could also be a part of it. But their study also had a lot bigger numbers. So this graph here actually shows this better, and what you see is that the only time we saw an increase in serotonin concentrations were when the 5-HTP was present. And we did not see an increase in serotonin concentrations with the negative DCAD alone. And again, there's a number of reasons why this could be, but it simply means that we're going to have to look at this in more detail, whether it's due to dietary component, or if it's due to sunlight, or if it's due to cow number or composition of parity. We're not quite sure yet. So this brought us to, is serotonin or calcium the cart or the horse? And they could be either. So this could work in one or two ways. We're trying to maintain our blood calcium concentrations. We're trying to keep milk calcium consistent. So one of two things could be happening. You see an increase in serotonin. This increases calcium flux into the mammary gland through these different pumps and channels I've talked about previously. And then this drives an increase in milk calcium, which leads to a decrease in blood calcium concentration, stimulating this bone turnover that we've talked about. Alternatively, it's this decrease in blood calcium because we have increased calcium transport into the mammary gland, increasing milk calcium concentrations. That could drive the increase in serotonin concentrations, and then this could increase the PTHRP effect on bone, or they could both be working. And we're not really quite sure where we are with that yet. Now, one study that we've done recently, and this is not published yet, was these were, and I will say that these are non-lactating, non-pregnant cows. So this is not a transition cow, but this is a dry cow that is not pregnant, so not lactating, non-pregnant. But the design of the study was that we were feeding the cows 12.7 kilos, and all of these cows did not leave any feed behind. So none of these cows had any weighbacks whatsoever. We were limit feeding them to make sure they were getting everything that we put in the feed. And all of these cows were fed a negative DCAD diet, as you can see here. But our question of interest was, how does dietary calcium affect this? So we had 3 levels of calcium. We were trying to target 0.41 and 1.6% of dry matter. What we ended up with was 0.45. That was the lowest we could get the diet, 1.13 and 2.02. And we did take wet chems on these samples for 28 days. So this is a very rigorous analysis of the diet. And that equals about 57, 144, or 257 grams per calcium the animals were getting per day. And there's 6 animals per treatment group. Now, the experimental timeline of this study was we had a 21-day feeding period. We did have to work on getting the diets in order prior to this, but once the diets were consistent and we knew they were getting what we wanted them to get, that was day 1 of treatment, and the cows were fed for 21 days. On day 19, we placed a jugular catheter, and on days 22, 23, and 24, we had 2 animals per dietary treatment getting EGTA infusions, which I'll explain in the next timeline. So these cows were receiving what we called an induction period or a hypocalcemia challenge, and we did this through giving them 5% EGTA, which is a selective calcium chelator. It's only going to bind calcium. And they were given this 5% EGTA at a constant rate of 500 milliliters per hour. So we would take a blood sample and we'd run an ionized calcium on them, and then we would start the infusion. And we blood sampled these cows every 15 minutes where we were measuring their ionized calcium concentrations. And we did this until they got to 60% of where they started from. So once their ionized calcium concentrations reached 60% of where they started, we then shut off the infusion. And we called this the recovery period. So they were no longer receiving EGTA. And we blood sampled at 0, 2.5, 5, 10, 15, and 30 minutes, and then every 30 minutes thereafter until cows reached 90% of where they started prior to the start of the infusion. So this blood value is 90% of where they were here. And I'm going to give you the summary of those results as we come forward. So we were trying to simulate a hypocalcemic event. And again, these aren't pregnant, and they're not lactating animals, and they're not a dry pregnant animal. They're not pregnant and not lactating. So we were trying to look at a model to control for as much as we could to see what the response would be if they were all on a similar diet except for dietary calcium concentration. During the feeding period, we saw no difference in rumination in these animals. So they were all eating the same amount, and as I said, they did not have any feed left behind, so the rumination times were similar. Their urine pHs were all below 6.0, but we did see that the low calcium cows had a lower urine pH than either the medium or the high calcium cows, but all of them were under 6. Now, this data is a summary of the ionized calcium concentrations, and this was during the 5% challenge period, so that middle timeline. The blue is the low calcium diet, the red is the medium calcium diet, and the green is the high calcium diet. On the x-axis, you have time in minutes of when we took our blood sample, and on the y-axis, you have ionized calcium concentration measured by the I-stat. And this break in the graph, I'll explain in detail as I go through this. So when we did the statistical analysis on this, what we were able to see was that to the left of this dotted line, which is at about 145 minutes within the infusion, the slopes of the graph of the different treatments are different. So the slope of the low, the medium, and the high are significantly different from each other on the left side of the graph. On the right side of the graph, they're all equal. But the high calcium cows have higher ionized calcium than either the low or the medium, and there's no difference between the low and the medium cows. Now, on the left side of the graph, though, all slopes of the lines are different. Now, the red line, or the medium cows, had the fastest rate of decline, or the steepest slope of all the treatments. The low calcium fed cows were intermediate, and the high calcium fed cows had the flattest slope of the three treatment groups, so to speak. So this just means that in response to the chelation of the calcium, or the induction of the challenge, that the high calcium cows were more resistant to the induction. So the medium calcium cows started to decrease much faster and much more rapidly, and the high calcium cows were much slower to respond. And in fact, you'll see in a second, the reason we only have this out to 360 minutes is because there weren't enough cows after this time point in the low and in the medium groups to make statistical comparisons to the high cows. And you'll see the average minutes in a second. Now, when we looked at the recovery period, and the hypothesis was actually that these high calcium cows would recover better to the challenge. But when you turn off the EGTA and you're no longer chelating the calcium, there's no difference whatsoever in amount of dietary calcium as to how they recover from the calcium challenge. And this just shows you that means that calcium is regulated very, very quickly and tightly. So the minute you don't have this extra drain on calcium or extra chelation and it's available, these cows will as fast as they can try to get back to normal. So when we look at total time to get to that 60%, so that 60% of where they started from during the chelation period, or the induction of hypocalcemia, it took the high calcium cows about 400 minutes once we started the EGTA infusions to get them to 60% prior to that initial blood sample. The low calcium cows were about 300 minutes, and the medium calcium cows were about 270 minutes on average to getting to that 60%. So you can see that this is it takes quite a bit longer to get these cows to that 60% of initial calcium concentration. In terms of grams, you do see that there was a treatment effect here too, and it took more grams, and this was corrected for body weight. So it just overall took more grams of calcium to chelate the high cows, and it didn't, and again, this was all corrected on a body weight basis for the animals. We did look at the serotonin concentrations during the challenge period as well, and what we found was that there was a treatment effect. And this gets back to this Carter-Horse question. So the low calcium cows had the lowest serotonin concentrations, the high calcium cows had intermediate ones, but these medium calcium cows had the highest serotonin concentrations. So this suggests to us that the cows, where they don't really know if they need calcium or not, or do they have enough calcium and they don't need to pull more from bone, or do they not have enough and they need to pull more from bone, they seem to be compensating as well by dumping more serotonin into their blood to potentially draw more calcium out as well. And so this was the first time we were able to see serotonin in response to a calcium. And it does indeed look like at least these dietary calcium concentrations, when we're chelating calcium in that hypocalcemia induction, that there is an effect on serotonin as well, and that we need to explore this cart and horse situation further. So we were really excited about this data as well. So in conclusion, we know that calcium is entering the mammary gland, and it's being actively pumped into the milk as well as being secreted along with protein. We also know that this can drive calcium sensing at the mammary gland, but serotonin might also be doing this directly, and we're not sure, is it both of them happening at the same time? Do they work in tandem? And we're working on that in experiments as we speak. And we know that this is driving PTHrP excretion from the mammary gland and production by the mammary gland. It might be the low calcium, it also might be the serotonin, or it might be both working together. And this is stimulating increased calcium resorption from bone to maintain or resume or bring a cow back to her normal calcium concentration during these early days in lactation. We know that serotonin is working directly on calcium, it's working directly on PTHRP, and it also might be working directly at the level of these different pumps within the mammary gland as well. So with that, I have to thank, of course, my sponsors, the USDA, and I've also been very fortunate to have good funding from industry, both from Phibro Animal Health and Arm & Hammer, as well as the USDA Hatch program at the University of Wisconsin, Cardoso Pharmaceuticals, and one of my grad students through the NSF. But I also have to thank my collaborator, Dr. Rupert Bruckmeyer, Dr. Bob Collier, Dr. Nelson Horstman, and of course, none of this is possible without all of these great students that I've had over the last 7 years that have worked really hard to produce this data, and I would not be at this point in my career without them. So with that, I'm happy to answer any questions that I'm able to do so.
Okay. Thanks very much, Laura. I am going to do a little bit of housekeeping before we get to the question and answer period. Please, if you have questions, either type them in the chat window or type them in the question and answer window. I'll see either place. Let's introduce our next webinar. We will be joined next month by Dr. Tom Overton, who's a professor of dairy management in the Department of Animal Science at Cornell University. Tom serves as the director of ProDairy and is the associate director of Cornell Cooperative Extension. Like most production-based professors, Tom wears many hats in his position at Cornell. He teaches the dairy cattle nutrition course for undergraduates, an upper-level seminar course for dairy-oriented undergraduate students, and co-teaches a course in dairy nutrition for veterinary students. He also works with students in the Cornell Dairy Fellows program. We are grateful he can take time for our webinar. This is going to be Tom's second presentation for us, and it's also our second sort of mini presentation on transition cows. His talk next month will be a focus on liver function. So please plan to attend April 11th at 6 p.m. Eastern Daylight Time. This will be one of the relatively rare webinars where the speaker and I are not only in the same time zone, but also in the same room. We've been told that maybe the whole staff will join us, so we'll try to keep the office shenanigans at a minimum for this. We, of course, could not do this webinar without the help from our really great co-hosts. We're joined normally by 3R Lab, Marcelo Ramos, for joining us, but he wasn't able to join this time. Paula Turiello in Argentina is joining us, and we want to give a thank you to Rock River Laboratory, who is helping sponsor her part of the webinar. We are joined this year for the first time by Tom Long from Hemingway, and he is translating the webinars into Mandarin. We also are very grateful to our generous sponsors who make it possible for us to get great speakers like Laura and manage the program. We thank our gold sponsors, Ajinomoto Heartland, Superior Nutrition through amino acids, makers of AgiPro L, and Arm & Hammer Animal Health, makers of cattle feed ingredients that optimize dairy cow health. Our silver sponsors are Dairyland Laboratories, Virtus, makers of Strata with EPA, DHA omega-3s, and Prequel with omega-6s, Cumberland Valley Analytical Services, Chemin, featuring USA Lysine, Dairy One Forage Laboratories, R&D Life Sciences, and AB Vista. Our bronze sponsors are AminoMax, Purdue Agribusiness, Jeffo, Quality Liquid Feeds, Adiseo, Origination Incorporated, and Novita. I'm going to open the floor up for questions. Because we have trouble with people's independent audios, we just ask that you type them in. Oh, good. Hi, Paula. I'm going to unmute Laura. Go ahead.
Hi. Hello,
Hi.
Laura. Great presentation.
Thank you.
I have a question from Pedro. Is the only way to administer 5-HTP is by injection?
That's a great question. So we have been doing that as of this point because we were unaware of what would actually happen in the rumen with the 5-HTP, to the cows at least. Our studies in rodents were done feeding, but we are right now actually analyzing a data set where we've put 5-HTP directly into the rumen, and we're working on our ability to determine if it gets past the rumen freely or if it's going to require some kind of chemical alteration, like, one of the amino acid type products. But we're analyzing that data right now, and it'll be presented at Dairy Science this summer. So we're hoping to feed it. That's the goal.
So Laura, I have a question on, I was also intrigued when I was reading the papers on how the serotonin would be administered on a practical sense on the farms. Do you have some insight on that?
That's great. So that's the stage we're at right at this minute. The end goal is to get it into a feeding-based product, because that's simply the easiest way to do something. And we, as I just mentioned, we've done a study where we put the 5-HTP into the rumen, and we're analyzing that data right now as to whether it gets out or not. And so we're hoping we won't have to rumen protect it. And once we figure that part out, then the next step would be to start feeding studies, knowing the rate of inclusion, how long we would need to do that, so that we can give better recommendations on that part. But I will say that starting, 6.5 now, almost 7 years ago, working on this at Madison, I thought it would take me 20 years to get to this point. So I feel like we're making pretty good progress, and I'm hoping we'll know in the next year or two, hopefully the next year, what that will look like.
I have a question from Debbie LaChapelle. She, first of all, says thank you so much for the presentation, and she was wondering if Jerseys responded in the same way as the Holsteins. I think you touched on that briefly, but didn't elaborate.
Right. It's a great question. We were able, that study that I showed that where we initially gave the 5-HTP to the prepartum cows, we were able to do a 2x2 factorial. So we had Jerseys on our farm as well as Holsteins here in Madison. And what we saw was that these Jersey cows, and this is published in that same paper where the reference is, but we saw that the Jersey cows, what happened with them when we gave them the 5-HTP prepartum, they started to see a decrease in their calcium prepartum. And once they hit parturition, they were already going up. They weren't going down. And so they seemed to behave a little bit differently. And a lot of things they did were inverse to what we saw with the Holstein cows, but at least preliminarily what we think is happening is they responded during the pregnancy stage, and by the time they calved, they were already on the increase in their total calcium concentrations, whereas even though the 5-HTP cows and the Holsteins, they were higher total calcium concentrations at parturition than the control cows, they still did see a dip postpartum as compared to the Jerseys. And I'd venture to say, and I've said this a few times, and I just need money to study Jerseys because people don't want to give us money to do that specifically. But I tend to believe that they behave a lot differently than Holsteins as a whole. And we saw that for a lot of metabolites, even in the control animals. And I don't think we've done enough research on them specifically to make sure that everything lines up just the same. And so it really would be worth doing so if we could get the funding to do that, especially because Jersey numbers are increasing in the United States, or they have been over the last few years.
Yeah, I know that there's a lot of pretty large farms that have some good numbers of Jerseys out in your area, actually.
Yeah, we just had one, I think, Milk Source opened like a 4,000 or 5,000 cow Jersey farm up near Green Bay, I believe. So they're there. They're not just
Question for you, second question from Debbie. Do you have any idea what the cost would be to feed 5-HTP, or are you just not there yet?
I don't have an actual dollar amount. Great question from her. But there's two things that I can tell you. We need far less 5-HTP than I ever originally anticipated, because what we were using in rodents was a lot more. But when you correct for metabolic body weight, that changes things quite a bit. So that's the one thing. We need far less than we would have ever thought. The other thing is that we've figured out how to make it without having to extract it from the plant that it comes from. And I think that's going to drastically reduce the cost. So while I can't give you a dollar number today, I think it's going to be feasible, and I don't think it's going to be outrageous. Otherwise, there's no way we would even try to do that. And myself and Dr. Horstman have the patent for this technology, and it's through Amelgo, which has also sponsored some of our research. And Dr. Bob Collier, my PhD advisor, and Nelson own that company, and they're working on developing this. And they've done a lot of the cost economic breakdown that they don't tell me a lot about. They just tell me to do science. So that's what I do. That's my best asset. But I think it's going to be manageable. And I'm hoping in the next year or two, we will have a better idea of that.
Okay, thanks. I'm going to let Paula ask a few questions. I know she's got some, and then I have some more in my window. A really interesting one that I think you'll enjoy. So, Paula, go ahead.
Yes. Another question is about serotonin. Is the greater calcium uptake by the mammary gland due to the higher serotonin level because of vasodilatation caused by serotonin?
That's a great question. So what I think you're saying is they want to know if the higher uptake of calcium by the mammary gland, is it due to vasodilation of the blood vessels that is also regulated by serotonin? Correct?
Yes.
Okay. That's true. It's true that serotonin does regulate vasodilation, but it also regulates vasoconstriction. And the one thing that I learned from the very beginning is that serotonin is inherently very complicated. It is possible that could be part of the situation, especially when you're giving 5-HTP, the precursor, at a whole systemic level. We believe, though, that what's happening, at least with the PTHRP and the calcium and the direction from the mammary gland, is partially due in response to serotonin produced by the mammary gland. And we have evidence of that in a mouse model where we were able to block serotonin production in the mammary gland only during lactation. And what we see is that about 50% of the serotonin concentrations in the circulation is decreased during lactation only. And while a lot of different tissues make serotonin, and largely the gut makes quite a bit, it looks like the mammary gland controls a good portion of this during lactation. But with serotonin, you can't rule out that might not be an additional reason why.
I have another one related to serotonin. Does serotonin also act on the kidney, reducing renal expression of calcium?
Does serotonin regulate calcium excretion from the kidney? I actually don't know the answer to that question, and it's a good one. We haven't actually looked at it ourselves. We've done some things that we haven't published where we've looked at like calcium transporters in the kidney of mice that are lactating, and we don't see the effects that we see in the mammary gland. Now, outside of lactation, I couldn't tell you anything because I personally haven't done that, but there is a possibility because we do know, at least in the gut, in non-lactating animals, if you stimulate serotonin somehow, you will see some differences in calcium transport, at least expression in the gut. So it is a possibility.
Okay. So this is from Jim Aldrich. What do you think the evolutionary advantage for lactating mammals is to have this mechanism? Why do mammary glands become somewhat of an accessory endocrine organ?
That's a great question, and the basis of my life, basically. What we do know, and a lot of like Dale Bauman's work would classically show this, and others, is that all mammals, once they start lactating, the mammary gland starts to make a lot of things, serotonin being one of them, and many others, that get secreted out into the maternal circulation and start to direct basically homeostasis. And in the Bauman and Curie paper from 1980, he talks about this in the adaptation to homeoresis. And so in this unique physiological state where the baby is reliant on the mother to stay alive and it's providing this, milk that is a complete food, you want that to take over. Just like during pregnancy, the fetus is really kind of pulling whatever it can. The baby is doing so during lactation. And so the mammary gland has to be able to tell the liver and in the bone and the muscle, like, you're not going to get what you're going to normally get because milk is king. And I have this, I was just saying earlier today, I was doing some teaching for the dairy producers of Wisconsin. We argue about this a lot, the nutritionist in our department and me, the lone lactation person. Is it the liver or is it the mammary gland? And I always remind them, if you cut the mammary gland off, there's no milk. But you can cut the liver out and you'll get milk for a little while at least. Not forever, but at least for a little while. And it's really because the body has to adapt to this higher physiological event. And this happens, Dale really shows this in a lot of different other situations, not just lactation, but like diving animals and in these very specific physiologies where something takes over the normal physiological event. And in the case of lactation, it's because you have to be able to make the milk to support the young, and that's why evolution created it.
So I was doing some looking through some of the literature, and the discussion of serotonin has seemed to, from what I could suss out, come sort of full circle. Initially, they thought that serotonin perhaps inhibited milk production, but then I was reading some of your more recent research, and serotonin is good, and it encourages milk production. What can you speak to on that?
That's a great question, and always like the $1 million question with us. But one thing we've learned about serotonin, and it'll show you, as I mentioned earlier, I think with Paula's question, is it the vasodilation? Well, serotonin also vasodilates and vasoconstricts blood vessels. So it depends on the concentration of serotonin, and it depends on the timing. And it's like that with a lot of hormones and a lot of different, metabolic endpoints. How much of it's there? What's the stage of the lactation cycle? Is it the transition period into lactation? Is it going into involution? Are you still removing the milk? And at high concentrations of serotonin, which would occur when you stop milking an animal, it'll get past the normal physiological concentration as it builds up within the tissue. And in that case, it'll cause the tight junction complexes to break down and accelerate dry off or involution. But if you're removing milk at a normal rate and you're in this normal serotonin concentration and you're lactating, there you have a positive effect, and it's affecting a different process. And this has a lot to do also with the serotonin receptor profiles in a tissue. So the mammary gland has anywhere from 3 to 5 serotonin receptors depending on what species you're in, and they all act differently. So like I said before, serotonin is a lot more, it's really complex, especially for something that's really a modified amino acid.
Mm-hmm. Let me see if Paula has any more questions. I'm actually going to check with Tom and see if he has any questions. I'm going to unmute him. Not sure how this will work.
Yes, my main question is, are there any people providing this commercially for the 5-HTP?
There aren't any companies right now, at least for cows, providing this commercially. You can buy 5-HTP on Amazon, at Costco, at GNC. Like, you can buy it anywhere, really. But Amelgo, which is the company that Dr. Collier and Dr. Horseman own, they're working on trying to get this out. Like I said, we have a patent, and they're working with some companies in the ag industry trying to move this to the next step. And so we're a lot closer than I ever thought we would be, and especially at the time in research where we are. So I'm hoping it will be in the next year or two that we'll have more answers about the commercial part of it.
Thank you. Some of the questions that I had from reading the Journal of Endocrinology articles and indicating that there's higher calcium levels in the milk with increased serotonin beyond is that what I understood correctly, beyond initial lactation?
That is correct.
Okay. And did you see any sort of significant difference between Jerseys and Holsteins in the calcium levels?
That's a great question. So in terms of calcium levels in the milk, definitely there's higher calcium in Jersey milk than there is in Holsteins. That was known before, published before. We know that they do that. We didn't really see an effect of the 5-HTP treatment on the Jersey cows as much on their milk as we did on the Holsteins. And again, we didn't have a huge N in that, but there seems to be some maybe like opposite effects that we would see on them. And I really think that Jerseys, there's a unique opportunity to try to understand them a little bit better in general, but also we would love to try to figure out how 5-HTP and serotonin really work in them specifically, more than what we have in that study. We just don't have the numbers or the funding support to look at that specific breed, but they are typically more calcium challenged, and that has to do with a couple things. They put more calcium into their milk. They have higher components, so there's going to be more calcium with the protein as well, but also they make more milk kind of on a per unit body mass base than a Holstein would. So there's some unique opportunities to really try to understand that from a biological perspective that I would love to do. if someone would like to really spend a lot of time studying Jerseys.
I'm a little curious, given the number of products that are offered to humans here in the United States that are fortified with calcium, is how significant is that difference, and is that some sort of is that something that should be paid attention to marketing-wise?
Right. That's a great question. And I think as we move more and more into these specialty milks, like the Fairlife milk, those are from Jersey cows.
I did not know that.
Yeah. And so, I mean, they do some other things to it, but it's largely from Jersey cows. And so I think that, we're going to have to start really paying attention to that, and especially because of the way milk prices are, like how can we make sense of what we have and maybe, manipulate some of these things. And one of the hardest things to manipulate is milk calcium. And so I had some people in food science like ask me about that, and they're like, wow, if we could really do this, that would be an added benefit because it helps the cheese processing effect. If you have higher calcium concentrations in the milk, they're better able to process it for cheese. I don't know a lot about processing for cheese, but that's what they told me. So being able to do stuff like that for milk, from fatty acids to minerals, I think this is going to become something we're going to have to pay attention to.
Good. Yeah. I know a little bit about processing cheese, and I can just tell you that we get about a 5% yield difference when we use Jersey milk compared to using Holstein milk in the same recipe.
Right.
So, let me see. I think that Paula doesn't have any more questions. I'm going to see if Tom does. Tom, are you good with questions, or do you have any more?
No, I don't have any more questions. Thank you.
Okay. Thank you very much for joining us. Without any more questions coming in, I think we have come to the point where we can call an end to this, just in case. Yeah. Well, Paula says if they have questions later, is it okay to send you some emails?
Absolutely.
Yeah. All right. Great. Well,
I probably will do the same.
Okay. Thank you, Tom. Thank you. So on behalf of my co-hosts who are joining me, I want to thank you, Laura. This has been really interesting for me. We talked a little bit about some of the human aspects of this, and I hope that everything goes well with your research and that you continue to get more funding in this area, because I think it's pretty important.
Thank you for having me, and thanks to everyone for joining us.
Well, yes, thank you everyone for joining. I just want to reiterate that we'll have this recorded and annotated so that you can hear it. It'll be up on the website in anywhere from a week to 3 weeks, seems to be my habit. And then we also turn it into podcasts. So if you go to the Ag Model Systems website, you'll find our archived webinars and our archived webinars turned into podcasts. So thank you very much, Laura. Paula and Tom, thanks for joining, and we look forward to seeing you in April on the 11th with Tom Overton, who will also talk about transition cows. Thanks again, everybody. Bye.